Hello, 231. We're talking about muscle tissue and we're doing the first part of muscle tissue. Okay. Sorry about my attire today. It was one of those days, which is fine because we are in COVID and now I'm in the slides. So we're just going to do the first part, we're doing the first part. It's always a lot, isn't it? Okay. So muscle tissue, it's not just muscle tissue. We're going to actually start doing muscle physiology. First of all, you should have your notes out and it says, what are the 3 types of muscle tissue? So you should be filling that in right now, right? Skeletal, and what's another one? Smooth. What's another one? Cardiac. Okay, and what are the functions? So you're about to see a bunch of functions. Oh my goodness, we have 600. We're not gonna learn all the 600. They move. There's a bunch of stuff I already put down here, so you don't need to write it down. They help stabilize our body position. Sphincters, oh my gosh, circular, my favorite. Circular muscles help us move. Just think about our whole GI tract is surrounded by sphincter muscle and helps move the food down. So lots and lots of functions. Also heat generation, which I am cold right now and I wish my skeletal muscle would do some heat production. So that's all we're gonna say for that. Okay, so a little bit more, the 3 kinds of details about the 3 kinds. All right, even though we're going to be focusing on skeletal muscle, you should still think about, are they striated or not striated? What is their function? Are they voluntary or not voluntary? Conscious, other information. So for skeletal muscle, it is striated and voluntary, right? It is, so you know, picking up this coffee is a choice, is voluntary. Other information, each cell is called a fiber. That's because the cell length runs the whole length of the muscle. So super specialized cells. Good. Smooth muscle, non-striated, right? Non-striated, non-voluntary or involuntary. Sphincters are an example of smooth muscle. Yay! Unconscious. Cardiac muscle is not voluntary, is striated. And they are autorhythmic. That means that they have their own little pacemaker. So your heart, cardiac muscle will continue, those cells will continue to contract, even if your brain's not connected. Ah, interesting. So before we go on, a few properties. They are electrically excitable. We're going to talk more and more about this, especially in the next part. But what that means is they respond to your brain. They respond to an electrical signal called an action potential. So there's these things called action potentials that travel down nerves from your brain, down nerves, down to your skeletal muscle. We're going to be talking about that a lot. They are, another property is they're contractible. They can contract. Sometimes they contract a lot, sometimes they contract just a little bit. And so followed by an action potential from your brain, followed by a signal from your brain, they contract. What does tension mean? So tension is kind of how much torque you produce, how much ability to move stuff did you make? All right, I'll never ask that question. But there's 2 kinds of contractions, isometric and isotonic. Isotonic, you might have heard of these before. Isometric means that, let's do isotonic first. So I have a weight in my hand and I lift it. That's an isotonic contraction. So you're actually moving that object. But let's say I put a car on the end of my hand and I'm going to try to do the same thing. That's an isometric contraction. So I mean, I'm contracting the muscles, but they're not able to move whatever that structure is. Both in both cases, you are contracting. Okay. It's extensible. You can stretch it without damage, and it's elastic. It returns to its original shape after contraction. These are all properties of muscle, and we're going to be focusing on skeletal muscle, but it still works for the other muscles too. Okay. So the next piece that we're going to be talking about are the wrapping around muscle, the wrapping, the organization around muscle. So yes, there are blood vessels and nerves. It's very vascular. But the connective tissue coverings are what we're going to really focus on. And so we're going to focus on these three types of connective tissues that are wrapping: endomysium, perimysium, and epimysium. And so the whole entire muscle is surrounded by epimysium. So it surrounds the whole muscle, and it's made out of dense irregular connective tissue. And yeah, that's great. The next wrapping, are groups of muscle fibers, and these are called fascicles, groups of muscle fibers. And this wrapping is called perimysium. So groups of muscle fibers are called peri. They're made out of dense irregular, and that's about it. All of them pretty much are dense irregular, except for endo, I think is areolar. But I won't ask you about endo. I might ask you about it. I won't ask you what kind of tissue it is. Okay. Okay. And then endomysium wraps individual muscle fibers. So individual fibers are wrapped with endomysium. Okay. So super strong. Tendons, what are tendons? Tendons connect the muscle to the bone. This is maybe the femur. And they are continuous with the epimysium. Okay, so aponeuroses are sheets of connective tissue, like on the palm of your hand. You have them on your stomach as well. So your rectus abdominis is connected to aponeurosis instead of connecting directly to bone. So when they're sheets of connective tissue. Okay, beautiful, beautiful. I'm moving on to page 3. Tendons, nerves, and blood supply. Let's see if we have something. Okay, I'm going to talk about that in a second, but tendons, nerves, and nerve supply. What is a tendon? Muscle to bone. What's an aponeurosis? Sheet of connective tissue that connects to the muscle. What are fascia? Fascia is usually wrapping the epimysium, and it connects to like fat or whatever tissue's next to it, okay? Fascia below the skin is what connects, it's usually connective tissue, but what connects that organ to the next organ, okay? Are musculars vascular or avascular? What do you think? Go ahead and circle it. Do they heal well? Why? Outline the beginning of life and muscle formation, including myosatellite. That's what slide we're on right now. What happens to these cells later in life? So we're on page 3. And so before you were born, there were these little stem cells. These little stem cells became muscle. But what happened is all these little muscle cells fused together into one big long cell. So there's a super specialized cell. And now it's no longer, if you tear it, it's no longer able to heal itself, except a few stem cells stick around. These are called myosatellite cells. And they are sitting around waiting. They're stem cells for muscle. They're sitting around waiting. So if you have a tear in your muscle, they can divide and become a new muscle fiber. Okay, so it is possible if you have a complete muscle tear to regenerate muscle. However, the older you get, guess what? The less myosatellite cells you have. Bad news. The older you get, the less of these. Okay? And I was right about the endomysium is areolar. I was right about that. Okay. We're done with page 3. On page 4, what you want to do is you want to take this list. There's a list of terms on page 3 in a little box, like sarcolemma, T-tubule, sarcoplasm, myofibrils. You want to take that list and make sure you can find it on this structure here. Okay, so on page 4, top of page 4. What are you looking at right there? What is that thing? Take a look at that. That is the cell of a muscle, of skeletal muscle, or that is a what? Fiber. Okay, so that's a cell, that's a fiber, that's a muscle fiber. And there's a lot of stuff in it. It doesn't look like the 112 little cell, does it? So you want to start, I personally would draw it myself, but you want to be able to find each one of these structures. So you have the outside of the cell, and you have a whole bunch of people that study muscles, so they have new names for everything. So the outside here is called the sarcolemma. There we go. Okay, outside is called the sarcolemma. The inside, instead of cytoplasm, it's sarcoplasm. We have mitochondria. It's good. We also have something called transverse tubules. Now let's see where they are in here. I think they're in blue here. Yeah. Okay, so transverse tubules invaginate inside the cell. So they're like little tunnels that go inside the fiber. Okay, and in here they're in blue. You can see they're kind of going through the whole cell, but they go from the outside of the cell, the sarcolemma, the outside of the cell, all the way in. So they're like little holes. You just think of them as like little holes. And they do have a special function. I'm going to show you some other things. This here is called a triad. So a triad has T-tubules. Remember, they're tunnels from the outside, has a couple T-tubules, and then in the middle of it, it has this thing called the terminal cisternae. Okay, that's in the pink or kind of purplish. Okay, that's called a triad. Good. Okay. Very good. Right now, we're just getting the anatomy. Trying to think of anything else we need to see. Oh, yeah. This is called a myofibril. Lots of these myofibrils, they're made out of proteins. Myofibrils. You can get more of these when you lift weights. You can get more myofibrils and make that fiber thicker. So these are myofibrils. And good. Okay. So you might also notice something else, that the myofibril is what's causing the striated look. Because look at these different colors. You can see something here, and there's a light, and there's a dark, okay? So myofibrils are made up of units. There can be hundreds of these all the way down the myofibril. Okay, myofibrils run the whole length of the muscle. But these are called sarcomeres. So a sarcomere is a very short little unit, and it's the thing that contracts. Okay, it's the thing that's going to cause contraction, cause that muscle. Muscles only get shorter, so they contract by getting shorter. And so you're going to have hundreds of these little sarcomeres all the way down the myofibril, and each one of those is going to contract, contract, contract. And that's how contraction happens, although we're going to do some details. But you can see that everything's kind of happening at this sarcomere. Okay. So I think we'll probably talk about sarcomeres next. Let's do it. Let's see what else. Okay. You might see the word myofilament, and those myofilaments are inside the myofibrils. So we'll talk about them, but it's basically, there's a whole bunch of proteins in there. Okay. And there's two types. I don't tend to use thin and thick filament. I tend to use the actual names of the proteins, like actin and myosin, and there's a couple more. So, but that's fine. You can use thin and thick. So thin filaments look thin under the microscope, and they're made of actin mostly. And then thick are mostly made of myosin, although not entirely, and they look thick. Okay. Cool. Let's see what's next. A couple more things about what you've seen. The Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum is ER. And what it does is it holds on to calcium. So the sarcoplasmic reticulum, it kind of looks lacy and blue. The sarcoplasmic reticulum holds on to calcium, okay? Holds on to calcium. All right. Good. We're not going to do physiology yet. We're getting there. We're not quite there. Okay. Now, what is a thin filament? Okay, what is a thin filament? Okay. So a thin filament is not just actin. So I would definitely draw this. We're supposed to be getting to the sarcomeres, but we will in a second. We'll get to a sarcomere in a second, okay? But we're going to look at these thin filaments first. So thin filament is composed of a couple things, all right? So these balls here, that's actin. These balls here are actin, okay? And another sort of strand, and they're kind of globular. There's a strand-like protein. You can see it's white, right? That strand-like protein, I'm going to outline in blue here. This strand-like protein is called tropomyosin. Super important. Tropomyosin. Super important. Okay, so we've got two proteins so far. Another protein is called troponin. Looks kind of like a shell, doesn't it? Troponin sits on the tropomyosin. Okay, and the actin sits on the tropomyosin. So the tropomyosin is like this big string that's holding troponin and actin. All right. What else do we want to say? I know there's some things that we're not putting in here. We will. Okay. Actin. Actin has kind of two sides to it. It has a side that's sticky, that sticks to things. And this is called the active site. I can't write it all out, but it's called the active site. And then it has another side of it that's not active, it's not sticky. So what you're looking at right now, the way that the tropomyosin is holding the actin, you can't see any active sites, so it's not sticky. So it's just sitting there and nothing can stick to it. We'll see that's important later. So the active site is covered. The active site of actin is covered right now. Okay, beautiful. All right, now let's talk about thick filaments. Let's talk about thick filaments. So thick filaments are mostly myosin. And what is myosin? Myosin's a protein, but it has a funny shape. It has kind of like a golf club head shape. Okay, so you can see lots of these little golf club heads. And these little golf club heads are really important. So this is a thick filament, has a whole bunch of these little myosin heads. All right, now what we're going to see later in another slide or so is we're going to see that the thin and the thick filaments are together in this thing called the sarcomere. Okay, this place where we contract, So thick filaments are in purple. You can see they've got the little heads, their little heads sticking up. And the thin filaments, remember, it has actin, tropomyosin, troponin, okay? And you can kind of see, you can kind of see that they're together. They're near each other. Look at that. They're near each other. And what's going to happen later, not right now, but what's going to happen later is for you to contract a muscle, they're going to have to get together, okay? And they're going to pull shorter. And we'll see how that works. So this is kind of jumping ahead, and we can always go back and discuss it. But what I wanted to do was get the anatomy first and then do the physiology. Okay, so a couple things to think about. Actin, thin, myosin, thick, are the proteins that are going to help us cause contraction. But we also need the string, tropomyosin, and that shell-looking thing, troponin. They also are going to help. We won't talk much about titin, but it's basically elastic, and it helps. Basically, it helps so that your muscle goes back to its original shape after you're done contracting. All right. Now, let's take a look and see if we've got a slide of a sarcomere. This is muscular dystrophy, but this is really what I wanted to see. Okay, so a sarcomere, that's actually better. Let's see if there's anything. Yeah, let's not worry about that one. Let's use this one. So this slide is showing you a sarcomere. Now remember, there's going to be a whole bunch of sarcomeres lined up all the way down a muscle, and that's called a myofibril, okay? So this is a sarcomere, and we're going to look at it from this is one sarcomere. The ends are called Z discs. I like to say a sarcomere is Z to shining Z, from Z to shining Z, because it's a Z disc. This is just a protein that kind of holds the whole thing together, okay? And then it's holding on to the thin filaments that are in orange here. And then there's an M line. This is the middle of this thing. And the M line is holding on to the thick filaments, or the myosin, okay? And you can see they're all kind of repeating. The green looking stuff right here, this green looking stuff, that's the titin. All right. All right. Now, this, what we're looking at is a sarcomere. We're looking at a sarcomere that has not contracted yet. We have not had contraction yet, right? So we've got this like relaxed sarcomere. What's going to happen is when contraction happens, we're going to pull the Z's in. We're going to pull these Z's in, and we're going to make this whole thing, these Z's, the Z's, we're going to make this whole sarcomere much shorter, okay? That's what we're going to do. We contract a muscle, we're making it a lot shorter, okay? Now, how does that happen? That will be the next step. So hold on, before you move on, let's make sure we answered everything on page 4. We have thin filament, myosin. Oh, zone of overlap, thank you. Zone of overlap is the overlap between thin and thick. You see it? Zone of overlap is wherever thin and thick overlap with each other. So when a muscle is contracted, you're going to have a lot of zone of overlap, because what's going to happen is the Z will come here. Z is going to move right here. The Z is going to go in this direction, and you'll have the orange extend into here. So you'll have a really short little sarcomere, but a lot of zone of overlap. Everybody see it? Zone of overlap. I think I should put that in the chat. What is the zone of overlap? Right? It's the overlap of the thin and thick, or the overlap between actin and myosin. Okay. Z discs, we said. For lab. Okay, so a couple lab things. The I band is wherever there's light. So this is the I band is right here. I band, this is all I band. I band is wherever there's light. I bands lack myosin. Okay, so they stain light. And then A bands stain dark. So it's wherever, this is an A band. A bands are wherever the myosin is. That's an A band. Okay. Good. I think we have labeled that. Oh, H zone. Sorry, I flipped over the page to page 5. H zone. This is for lab. This is the H zone right here. H zone. Middle of the A band, only thin filaments, okay? Only thick filaments is the H zone. The M line is the very middle of your sarcomere. And the middle of the sarcomere does support myosin. Okay, we said that. Good. I think we're good. Now hold on, did we just contract? Oh no, this one just shows us all the different zones of overlap and everything, okay? All right, so we didn't get to see a contracted sarcomere, but you can imagine it would be very short, right? We move the Z on both sides. We'd have a short sarcomere. That would be contracted. So every time you contract a muscle, you did that. Isn't that crazy? Every time you did a contraction. I thought I moved this, but I actually don't. I don't want to add this in yet. Yes. I don't want to add this in yet. Okay. All right. So we're going to actually go back for a second. I want to go to a place where there's not much on the slide. Just stay with me for a second. Yeah, I'm just going to go here. Okay. I want you to imagine that you want to pick up a soda. Okay. You want to pick up a drink. You want to pick up a soda. So your brain, there's neurons in your brain that are going to say, I'm going to contract the muscles of my hand, okay, so that we can pick up the drink. I'm also going to contract some other muscles, but let's just focus on one muscle. So I'm going to contract that muscle. Well, how's that going to happen? Well, I'm going to send a signal from my brain down my spinal cord, out of my spinal cord, out to the muscle, to a specific muscle fiber, cause that specific muscle fiber to get excited and contract sarcomeres, okay? That's the big picture. And I've actually had students, for me, I actually drew that out because we're going to learn every single piece, the details of every single piece of what I just said for the next 3 chapters. The motor neuron, what does it do? Excuse me, the neuron up here, what is it doing? Sending an action potential, sending a signal. How does it travel? Where does it go? Down to the spinal cord, out of the spinal cord, till it gets to a skeletal muscle fiber, causing that skeletal muscle fiber to get excited. What happens inside of it? How does the sarcomere contract? That's what we're going to be doing. But today, the only part we're going to do today is this part right here, the part where we make that sarcomere short. So the entire process, starting with the nervous system, is long. Okay, and we just did the visual of it. Okay, we're only going to be doing this part right here. And relaxation really is just sort of everything lets go. So we're going to do this part 3, and part 4 we're never really going to do. So we're going to do part 3. That's contraction, which means that's the shortening of sarcomeres. That's the shortening of sarcomeres. And we're going to use calcium and ATP. Okay, so calcium and ATP are essential for muscle contraction. I bet you didn't know that. Maybe you do. Maybe you did. Okay, and I'm not worried about this word, but we're going to talk about sarcomere shortening, which is the same thing as sliding filament. So you can use either word. All right, so you should be on page 5. I'm actually not going to do any of this. I'm skipping over this. We will do all of this. This is the nerve. This is the nerve and the neuron. Okay. Actually, we're not doing that part either. Aha, here we go. We are going to be looking at, what are we going to be looking at? We're going to be looking in the sarcomere. So you have a sarcomere, right? And inside a sarcomere, you have the thin filaments and you have the thick filaments, okay? So the thick filaments is myosin, mostly. The thin filament has the tropomyosin. It has, that's the stringy looking thing. It has the troponin, which is this shell, the yellow thing, and then it has actin, which are the balls, okay? Okay, very good, very good. All right. And I would, it says outline flow or draw the contraction cycle. Okay, so the first thing we want to think about, and I would actually draw this. My personal thought would be I would draw this, but it's okay. What we want to do is we want to show, you can draw this as a sarcomere if you want. You could draw your Z, and you have your Z over here, and you have your H, or the middle line, sorry, you have some thick filaments, those are myosins. And then you can draw your little actins in. So you could do this if you want to, just like this, okay? As we kind of go through this process, all right? So what's gonna happen is you'll be sending this signal from your brain, which we'll fill in later, but eventually it makes it to the skeletal muscle fiber. And eventually calcium is gonna get dumped onto our sarcomere. Now, we'll fill in a little bit more. It is the sarcoplasmic reticulum that does it, but I'm not that worried about it. What I want you to first see is that calcium gets dumped onto my sarcomere. Calcium gets dumped onto my sarcomere. Okay, calcium gets dumped onto my sarcomere. Calcium gets dumped on my sarcomere. Well, somebody really likes to catch calcium. Who is that somebody? That somebody is called troponin. So it's that little one that's like the little shell. It loves to catch calcium. So you can kind of think of troponin as like having like a little catcher's mitt. Like I like to think of troponin like, here's troponin, it has a little catcher's mitt. When calcium gets dumped, it grabs onto it, okay? Who does? Troponin. So troponin grabs onto calcium. As soon as that happens, troponin will cause, and it's holding on to tropomyosin, that big long string, right? So troponin will just like roll this way, like this. And when troponin rolls like this, it's going to cause that big string, tropomyosin, to move. And when tropomyosin moves, it's a big long string. All of those active sites, the sticky parts, are going to be exposed. All of those active sites, the sticky parts, are going to be exposed. Okay? So, what does that mean? What does that mean? They're sticky now. So now they're sticky. So you can even, on your drawing, you could even have like, they're sticky now, they're sticky. All these little actin are sticky. Well, somebody actually really likes it when they're sticky. Myosin does. So myosin has these heads, right? We said they look like golf club heads. They're always sort of sitting like this, right? They're always sort of just hanging out. As soon as actin's sticky, guess what the myosin's going to do? Grab on, grab on, grab on, grab on. This grab on, okay, this grab on is called a cross bridge. This grab on, myosin grabs on to actin. When this happens, this is called a cross bridge, okay? And it is in your notes. It says attachment of myosin head to actin to form cross bridge. Now the energized myosin, okay, so it's got a cross bridge, right? Now the myosin is going to do what? It's going to, on the end of the myosin head, it's holding on to an ADP. See this little ADP? It's holding on to it. Okay, so what's going to happen next is it's going to release ADP. It's going to release the ADP. So boop, off ADP goes, and then because of that ADP chain leaving, it will cause it to pull. So that head is going to pivot and pull. Okay, now, so everybody stay with me. And these are all, you've got like, let's say 600 of these heads pulling. They're all, it's like a tug of war. They're all pulling. Why? First they form the cross bridge, then it's called the power stroke. ADP leaves and they go like this. ADP leaves and they pull, okay? So I'm gonna draw that here. So I'm gonna draw that here, over here on our other side. Okay, I just wanna take that off, okay. So we formed the cross bridge, but now we need to form the power stroke. So what's gonna happen is these guys are gonna move this way and they're gonna pull. They're gonna pull, they're gonna pull, they're all pulling. And what is that gonna do to the length of the sarcomere? It's gonna make it shorter. It's gonna pull the filaments in, and now the Z line is gonna be over here. Okay, so we just did it, power stroke, pulled everything in closer. Who did the power stroke? Myosin. Okay, so we just did the power stroke. I'm not going to track phosphate, but I am tracking the ADP. So we did, power stroke was the loss of ADP. Okay, now we're still holding on. We're in contraction right now, right? So the only way to let go, the only way myosin is going to let go is if we have another ATP. So ATP is going to come and bind, and I'm going to use ATP as purple. So ATP is going to come and bind to these myosin heads, okay? When ATP binds to the myosin heads, they will let go. they will let go. Oops. They will let go. So they're going to let go. They're going to let go. All of them are going to let go. And what's going to happen to the sarcomere? It'll go back to the original length. Okay, they're going to let go. Now, when they let go, they've got that ATP on them. They've got the ATP on them, and they look really floppy at the moment. They've got, their head looks kind of like this. They don't look like golf club head. So what'll happen is the golf club head will take the ATP and split it and just keep a DP on it. And it's called cocking the head. It'll make it so it's shaped just like this. And I'll show you that in the slides. Okay, now it's back and ready for the next contraction. So I like to draw it as a sarcomere, but you can also draw it the way that they have in the book. That's fine too, okay? It's really helpful to draw this. Okay, so this is, we've let go. ATP has bound to my little head. Everybody see it? We let go. We're going to recock the head. That might be on the next slide. No, it's not. No problem. We're going to recock the head by splitting the ATP, and we're ready for the next contraction cycle. Now, rigor mortis is when there's no more ATP. So what'll end up happening is the myosin will just stay held on to actin. All right, I would like everybody to go over the steps of this contraction of the myosin and bring it to class with you. Maybe you drew it, maybe you wrote it out. I would like you to bring this to class. Please bring it to class. We really need to go over it. Okay, so bring it with you to class. You can take a picture of it. You can put it into the chat. You can send an email to me, but please bring this to class so I can see your understanding, because this is really important physiology. Okay, we're getting into physiology, and so I really do need to see your example. So if you draw it, make a picture of it, that's great. If you write it out, that's great. But maybe I'll make a little document, a little Google Doc. I probably will. I'll probably make a little Google Doc so you can just put it up in there. But otherwise, send me an email with it, please, or bring it to class, every single person. Okay, that's the hardest part of today. That's the hardest part of today. So, what's the next part? So I wanted to show you this. This basically shows you what it looks like when you're not contracting, you're relaxed, and when you are contracting. So all your sarcomeres, you can see the zone of overlap is extreme here. Whereas when you're relaxed, there's not much zone of overlap. There's a little bit. Okay. So when myosin cross bridges are activated, anyway, this kind of talks about how when you're contracting, when myosin pulls in, the zone of overlap is great. It increases. Okay. Beautiful. Oh, this is really what we wanted to see. This is great. Okay, this might be better to use, okay? So we can see, here's your little sarcomere. And what we've got here is we've got the thick filament, the thin filament, and we've got cross bridge formation right here, right? So calcium gets dumped onto troponin, tropomyosin rolls, actin is active site, sticky site out. Cross bridge formation, this is the power stroke where myosin pulls in and releases ADP. ATP reattaches here, myosin lets go. And we can go back to another contraction cycle, okay? So we did it really fast, and I'd like to see this. I'd like to see you do this, please. Don't worry about this slide, because this slide is what we'll move on to the next chapter, or sorry, not next chapter, the next part of this video is we'll start talking about what happens in the whole muscle fiber, and then we'll work towards our brain. But you're welcome to go on and look at it if you'd like. Oops, having trouble. Okay. Okay, Last little piece of this chapter is the bottom of page 6, talks about length tension relationship. So, you know, when somebody's moving out of their house and they're like, can you, or they're wherever they live, can you help me, you know, move boxes out of my house because I need help. And you're like, okay, yeah, I can help you. And so you go and then you're going to pick up a box. But everybody says, don't lift with your legs, not with your back. Everybody tells you that. Don't lift with your back because those muscles are not strong enough. Now, what this has to do with is this length tension relationship. Don't worry about this graph too much, okay? Unless you like graphs. But basically what this is saying is when my muscle is relaxed, my sarcomeres when my sarcomeres look like this, where I have a decent zone of overlap, this is the myosin. When I have a decent zone of overlap, this is the myosin, and here's the actin. When I have a decent amount of zone of overlap, when my muscles are relaxed, like in my arm, there's a decent amount of overlap, okay? So this is a relaxed biceps muscle, okay? So I reach down with my legs, right? I bend down with my legs and I pull up with my biceps. I have enough zone of overlap to really cause tension. I have enough myosin heads to really cause tension and everything's fine, okay? So this is kind of the maximal amount when I have a medium amount of zone of overlap. Now, when I'm already contracted, I can't contract more because I don't have any more zone of overlap. So when I'm already contracted, so if I look like this, and here's my actin, if I look like this and I'm already contracted, I've got nowhere to contract to. So once you've used your bicep muscle and you're all the way up here, you can't pull more, you can't contract more because you're already at the top of your head. There's nowhere to go. Your sarcomeres are shortened. So that's what's showing over here. Okay, so you don't have much ability to produce tension or to produce torque. The other direction is what people generally don't think about. When you're reaching down, if you use your back muscles, when you're reaching down to grab that off the ground, here's your discs, here's your Z discs, and here's your M line, and here's the bands. Okay, those are my thick filaments, and here's my thin filaments, and they have very little overlap, right? Just a tiny bit of overlap, just teeny tiny bit of overlap because you're stretching down, right? You're stretching those muscles so they're stretched. So the sarcomeres are all stretched out. How much overlap do I have? Hardly any. So how many of those little M's, how many of those little myosin heads can pull? Not very many. So what will happen is you'll end up pulling those sarcomeres. And then you grab a box, you'll end up trying to pull those sarcomeres and they have very little overlap and you'll end up ripping them. Or you can rip a whole bunch of your sarcomeres. When you rip, it's okay to rip some sarcomeres. If you rip a whole bunch of sarcomeres and you rip that muscle, you will be laying down and your back will hurt for a long time. So that's showing over here, you don't want to have your muscle too stretched out. Okay, that's what this whole thing shows. You don't have to interpret this graph, but you do have to understand how our sarcomeres work. Okay, that's that last length tension relationship. All right, we are done. There's lots more slides to look at if you'd like, but we are done with this first packet. Lots and lots of stuff to cover in this chapter. But we are done with that first packet. Take a break and watch the next video when you're ready.